Okay, thank you, Kubo, for introduction. And I'm happy to be here to tell you something about biodiversity ecosystem functioning research, but also the same thing from the other side. I was working on this for my whole PhD study and few months after and few years before. So this is basically what I've been doing all my scientific life. Starting a little bit broadly, we will focus on two perspectives, how to look at the relationship between diversity and productivity. First, we will try to predict diversity by productivity, and I will try to mark this throughout the whole presentation by this blue rectangle. This is usually done in observational studies where scientists look at effect of productivity on diversity, and typically if you are on some productivity gradient, for example, increasing productivity, you are somehow changing the coexistence mechanism and it's leading to some changes in biodiversity. However, it's hard to say if this relationship is causal because it's just observational data and you don't know what's affecting what and what are the other variables in this relationship. The other possible view is to look how diversity affects productivity. So just swapping these two variables and asking similar question. This field study, we can call it biodiversity ecosystem functioning research, and this is typically based on manipulative greenhouse experiments, field studies, but typically with some created gradient of diversity, manipulated diversity, and here you can more precisely talk about some causal relationship. Okay, so that's two big topics of this presentation, but what I mean by the terms I used, what functioning means. Typically, it's just above ground productivity, and more specifically, it's just above ground biomass. Most of the studies are using above ground biomass as some surrogate for functioning for one function. However, even above ground biomass can have many meanings. It can be some proxy variable for other things like, let's say, photosynthetic capacity of community and so on and so on. So it's again just some proxy variable. So most of the talk we will look at above ground biomass. Also, one part will be about below ground biomass, which is very understudied. We will partly look at stability of above ground biomass in time, so how the biomass fluctuates in time, and what I mean by diversity, I am mixing terms, always calling it biodiversity, diversity, so whatever I say, I mostly mean taxonomical diversity, so just counting of species, 1, 2, 3, 4, 5, 6 species, and so on. And we will slightly touch also the functional aspect of the diversity at some point. So first part, predicting diversity by productivity. In theory, we could expect something like this, some unimodal hump-shaped model where this is theoretical, this is more from real observational studies, and this is typical for temperate grasslands, but it has been described by many authors across the world. The theory behind is that if you are going from low productivity, here it's productivity or biomass, from low productivity to medium productivity levels, the biodiversity on the y-axis is increasing simply because there is more resources, species can utilize more of these resources, so the communities can contain more species. However, from medium to high productivity levels, as the biomass grows, especially competition for light starts to play bigger role, which is subsequently leading to loss of species, because species that can't withstand this, let's say, hard competitive pressure will disappear. The problem is that you can find whatever you want. You can find positive relationship, no relationship, negative relationship, this unimodal relationship, and it differs if you look at different scales, if you look at different productivity estimates, if you do it in different habitat types, if you do it in temperate or tropical regions. Basically, wherever you look, you find something different. So, if you look in temperate grasslands, which is also where I've been working, mostly you find something I have already shown you, either negative relationship on this part in more productive meadows or unimodal relationship. Usually this is based on estimates of above ground biomass. It's usually done on rather small spatial scales and smaller extents, so smaller areas. On the other hand, if you look into tropics, it's generally done in forests, so the estimates of productivity are also different because it's hard to harvest forests, so there are some indirect measures like precipitation and DBH, which is nicely summarized here in Šimování Štorch 2017. It's usually done on bigger scale, bigger extent, and you typically get some positive relationship. Please ignore that axes are here swapped. It's very common. No one knows which direction it should go, so you can find both ways. Anyway, in tropics, it's typically positive. However, there is an issue with how to summarize these all varying relationships together. So there has been several attempts, several meta-analyses putting different studies together, merging studies from different scales, extents, from different regions, using different productivity estimates, and it has been also hugely criticized. One of the first big meta-analysis of this relationship was done by Mittelbach 2001, who showed that basically most of the relationships are humpback. The relationship I have shown at the beginning, some positive and so on, but most of them were humpback. However, there were a critique of this meta-analysis by Whittaker, who said basically Mittelbach did it wrong, because they merged different scales, as I have said, different productivity estimates. Their statistical approach was kind of weird, using significance level on 10%, which is not very typical. They didn't control for or over dispersion, and so on, excluded some outliers. So Mittelbach responded, okay, you are right, we did it wrong. However, you also we just excluded outliers, you excluded all studies. The results are fine, just with few mistakes. So then there was another analysis of the very same data. So they looked at the same data and said, okay, Mittelbach did it wrong. It depends, all of it depends on scale. Out of 150 studies, we used just 60 because the rest was not worth using. Then there was another big meta-analysis of the same data saying Mittelbach did it wrong. Tropical regions, positive relationship, temperate zone, unimodal. There was, again, Robert Whittaker criticizing basically all of the previous critiques of the original paper, saying Gilman and Wright did it wrong, and Partle et al. Did it wrong because they included some data sets twice. And his basically conclusion was, if the data aren't appropriate for meta-analysis, it's invalid to proceed with one. Then there was another reanalysis of the same data saying that productivity is actually a poor predictor of plant species richness, and that, and again, response that these guys did it wrong because they removed some studies to get no relationship at all. Then you have already seen this. This is not a meta-analysis of the same data, but a collection of new data across the world showing, okay, finally we have something to rely on, because this was newly collected data with the same methodology across whole world, so we can trust this. Okay, another reaction, they did it also wrong, some statistical issues and so on. I don't. If you were expecting, I'll tell you how it's supposed to be. I don't know. I just want to point out that a lot of people just are looking at this relationship, trying to analyze the data, and getting completely opposing results from the very same data. So, and I don't know how to do it better. I just want to point out that it's not very clear what's the relationship. So to increase slightly the confusion, we decided to add one more study trying to look at this relationship and check if it's consistent across different regions, different spatial scales, while using various productivity estimates, and doing this with exactly the same methodology, so not having the problems with comparability as it's in the meta-analysis. So what we did, we counted species at 3 spatial scales, did some estimates of productivity via above ground biomass, below ground biomass. We used several soil fertility measures, pH, which affects nutrient uptake, and did it in 2 regions, around České Budějovice and in Železné Hory, on 52 localities. And what did we find? Please don't try to go into detail. What's important is each graph is different estimate or productivity measure. Different colors are different regions. Red one is České Budějovice, blue, green, or what's the color is Železné Hory, and in black it's combining these 2 data sets. So if we zoom just, let's say, to 2 mostly commonly used characteristics, here you have above ground biomass, below ground biomass. You see that it's consistent across spatial scales, so the relationship stays the same. We haven't found any unimodular relationship at all. Everything was linear, either negative, positive, or none. For above ground biomass, you see that in Budějovice it's negative relationship. Good, we are in the relatively high productivity levels. So from the unimodular curve, it's the decreasing part, fine. In Železné Hory, the same. The issue is that when you combine these data sets together, as it's done in the meta-analysis and many studies putting data together, you get completely opposing relationship to what you see within the region. With below ground biomass, it's a different story. In Budějovice, you have somehow positive relationship. In Železné Hory, negative. If you put it together, it's positive. So if you merge the data sets, it's providing conflicting results. So the take-home message from this part can be that species richness productivity relationship is hugely context dependent, and if you are putting data together, you should be really careful, because there are probably different mechanisms playing a role in different regions. So we should be really careful when analyzing data together, and we did it in temperate grasslands, so we should be cautious with generalization of these results, but I don't think we would get more coherent results when we would expand to other habitats. So, with this disappointment, we can move to another, let's say, chapter where we just swap this relationship and look how diversity predicts productivity. And as I have said, it's called biodiversity ecosystem functioning research. Well, why to do this? Maybe because people always wanted not just to observe patterns, but they wanted to species not only to be beautiful, but also to be very useful. So different people predicted different, let's say, ecosystem services or functions by species diversity, showing that diversity matters for some supporting services like nutrient cycling. It's higher diversity is providing us food, it's regulating climate, it has cultural, aesthetical aspects, and so on and so on. Basically, the diversity is driving every function in community or ecosystem you look at. So the basic question here is if this nice species-rich community is functioning, whatever it means, better than this ugly species poor community. But in most cases, as I have said, the functioning basically means above ground biomass or above ground productivity. So theoretically, we would expect something like this. If you are increasing number of species, the function here, productivity, is also increasing. But the relationship is not linear, it's increasing but saturating because there is some overlap between individual species functioning. This is typically explained by two mechanisms, two theories. One is complementarity effect, which predicts that if you are adding more and more species into community, each of these species can somehow utilize the resources, has some is somehow using the available niche space, and if you have more of them, then the total used niche space or and therefore the function is also increasing. So if you have more species, they are functioning better, but of course there are some overlaps with how they perform, so it's not linear but increasing, saturating relationship. The other explanation is selection effect, or formerly called sampling effect, which is basically mathematical necessity, because if you are increasing number of species in community, you are increasing the chance of including highly productive species or species which is doing most of the functioning, and simply you get better functioning because the chance of having their better functioning species is increasing if you are selecting from more species. There is a lot of huge biodiversity experiments in Indiana, Cedar Creek in Minnesota, some ecotron experiments, and more and more. This is a wrong picture, which looked at the effect of biodiversity on various ecosystem functions. And what they did find? Well, if you are increasing species diversity, you see exactly what's in theory. You see increase in community biomass. And if you look in a longer time scale, you see here that at the beginning of the experiment, the differences between low diversity and high diversity plots are relatively small, but with increasing time, the difference is getting bigger and bigger, telling us that in time the biodiversity effect is getting stronger. So more species, better, higher above ground biomass, higher productivity, and it's strengthening with time. This is another example from different biodiversity experiments saying exactly the same. With higher number of species, the above ground biomass is increasing, but saturating at some point. And this is not just above ground biomass, but total biomass, so including below ground biomass. Again, basically the same relationship. The issue with these experiments can be, and it has been criticized by several people, including Shuspa already 20 years ago, saying that the community composition of these communities is artificial because it's usually sown from some predefined species pool, and the individual communities are assembled basically by a random selection, thus creating unrealistic species assemblages. And there were other people being stronger than Shuspa, saying that everything that has been done before 2019 is not even wrong, it's even worse than wrong. Or David Wardle was suggesting that we should more focus on some biodiversity experiments in real-world communities where we can make a stronger link between the theory and the real effects of biodiversity on productivity, basically. And this is a nice review and meta-analysis testing whether theory here it's grasslands here for above ground biomass, but they look at many other habitats and many other functions, but we are interested in this. Theoretical relationship is positive, that's what I have told you in experiments, that's what you have seen the slide before. Also, results are congruent with theory, showing a positive relationship. However, if you look into non-experimental studies, some observational studies, and not experiments, just observational studies, you see something very different. There are some positive relationships, but most of them is basically none. So therefore, we decided to create a relatively small biodiversity experiment on Shuspa's favorite locality Ohrazení, where we created diversity gradient, creating their plots with 1 species, 3 species, 6 species, 12 species, and then we had 2 controls. We had some baseline data in 2016, biomasses of individual species. We sorted them like this and created monocultures by removal of all species except of the most dominant species in that particular plot. If we wanted to create 3 species plots, we just took first 3 dominant, so the composition in each plot was different, and this was how we created the diversity gradient. Then we had 2 controls, 1 intact control, nothing done with this, and 1 disturbance control, because we were slightly scared that by manual weeding of individuals and individual species from plots will cause some effects by disturbing the plots, which will somehow mask what the patterns we want to see. So we had there also disturbance control, where we somehow intermediately disturb this plot without taking out specific species, just taking individuals pseudo-randomly, let's say, to control for unwanted effect of disturbance. And for purposes of this talk, we took above ground biomass of all species and measured also some traits and asked a very simple question: if species diversity is a good predictor of community productivity assessed via biomass. We also asked if dominant species are able to compensate for the species loss, if 1 dominant can compensate for a loss of other 40 species, because it's very species-rich community, and if there are some indirect drivers influencing this relationship. So this is how monoculture can look like at the beginning of experiment. This is how it looks a year after starting of the experiment. This is how many 4 years after the start, and I didn't find recent photos. And this is basically the simple result. If you look at the year as effective species richness, but it doesn't matter what you put there. You see no relationship with productivity, so it's not corresponding with theory. If we have more species, it's producing exactly the same biomass as a single dominant species, and basically the same information you see here. It's just divided into individual treatments, monocultures, free species, and so on, and controls. And across the treatments, you see no significant difference. And there is just some variability between individual years, which is not surprising. What's maybe worth mentioning is that the monocultures, although they are on average producing the same amount of biomass, they seem to be slightly more variable. And this is a structural equation model looking at the same thing, how effective species richness is affecting above ground biomass, but via different things. We were specifically interested if this disturbance is doing something with our communities, and it's not. So, of course, effective species richness is affecting disturbance because it's causing it, but it doesn't have any effect on functional diversity or above ground biomass. But there is an indirect link from species diversity to above ground biomass via functional diversity. So apparently there is some, although weak relationship, but we can link it more to functional, some plant characteristics rather than to diversity itself. So take-home message here is that unlike in traditional biodiversity experiments, species richness doesn't matter so much, at least for above ground productivity, and it's basically enough to have single dominant species to compensate for the rest of the community. So above ground nothing. So let's check what's happening below ground. Theory is basically the same. If you have more species, everything is better. This is a study of Ravenek from 2014 showing exactly what theory predicts, positive effect of diversity on below ground productivity as well as above ground productivity, and it's across many years. Also, you have here above ground part, below ground part, diversity gradient, and you see that at the beginning of their experiment, it's again IANA experiment, this I call it artificial experiment, where at the beginning there was no relationship at all, after 1 year no relationship at all, but after how many, 3 years, it started to show something, and after 8 years they saw already very strong positive relationships, so they had to wait for a while, but finally they found this relationship also below ground. So the issue with below ground is that it's very complex. There are maybe different drivers of the relationship, like fungi, bacteria, which is what this nice study of Wag et al. 2021 showed, that if you have a higher diversity of bacteria, you have also higher diversity of plants and higher productivity, and the same holds for fungi. So there are apparently some loops, some cycles. So we don't know if the changes in diversity are driving above ground biomass. Here it's actually stability of above ground biomass, but for the biomass it's basically the same. So we decided to deal with this issue simply by completely ignoring this and just looking at the effect of diversity on productivity without concerning this. So in exactly the same experiment, we looked below ground, we did 2 types of coring, one for standing root biomass and one in-growth cores, where we looked at the resilience of the community, but I don't gonna I'm not going to talk about resilience, so we'll just look at, again, the simplest possible link between diversity and total productivity. We cored 40 centimeters cores, divided then into different layers, washed roots, sorted them into rhizomes, tree roots, and other roots, and also scanned them because we wanted to look at some traits also. So this is the graph you have already seen. I have put it here so you can compare it with our results, which are done in the same style. So above ground biomass, below ground biomass, you have seen already above ground biomass in the previous slides that there is no relationship at all with. Here it's not diversity, it's different treatments, but it's sorted, so the diversity is increasing from left to the right, and what you see below ground is, again, basically nothing. There is some decrease in total productivity in time, but no differences between individual treatments. One can say that there wasn't enough time to differentiate, as here it's how many, 6 years, so maybe we'll see positive relationship in future, but we don't see it now, again, as with above ground productivity. So why there are such contrasting results between observational studies, the first part I've been talking about, and these artificial biodiversity experiments? There can be, and first I have to say I don't know, but we can speculate about potential causes mechanisms. Well, one thing is that it's done on different scales. The observational studies I've been talking about at the beginning are usually done at a region or sometimes even landscape scale, big scales, while biodiversity experiments are usually one locality and small, usually relatively small plots. In observational studies, there is often different metrics for productivity, while in BF experiments, it's basically just above ground biomass. This can be maybe nicely seen here. Bartlett et al. 2007, which was criticized by the other authors, if you remember, but doesn't matter now. They showed that with increasing latitude, there is a change between the relationships. So he's showing that there is basically less unimodal relationships in tropics and more unimodal in temperate zone, and vice versa for positive. So more positive in tropics, and we can simply link it with different productivity estimates used. Here it's biomass, here it's NDVI, precipitation and so on. Here it's biomass. Maybe it's just that these two approaches are asking different questions, because and it was also criticized that BF experiments are not studying role of biodiversity itself, but rather effect of species pool. We will get there soon. Or there is very nice recent study, 3 years old, from Hagan et al., showing that we maybe don't need to necessarily expect positive relationship in BF experiments, because again, these two approaches are looking on the same thing, just using different methodology, using different things, and he's saying that basically if we would use, instead of diversity species pool, it would be the same. Who knows that this remains to be somehow tested. Or the whole thing can be just because we are not able to properly analyze data, I don't know. And now, why such contrasting results within the universe of BF experiments? So the artificial BF experiments and something in a real community, something what we were trying to do somehow. Well, difference between this and this, basically, why? One thing, and I have already mentioned this, can be unrealistic assemblages, because biodiversity experiments are assembled usually from some species pool. You have, let's say, 60 species and create individual communities with different diversity levels just by random picking of these species and putting them together randomly. Nice example is this study from 94, I think, of Naim et al., where they created low diversity communities with Senecio vulgaris and Stellaria media. Fine. Then they created medium diversity communities by adding Chenopodium album, which is, you know, how Chenopodium album looks like. So no big surprise that the productivity increased if you increase diversity by adding Chenopodium album. And then they had high diversity communities where they included some other funny species like Sonchus coloratus or even invasive species like Conisa canadensis. Again, not a big surprise, the productivity increased. There was a nice study of Romanuk et al., 2009, where they, although it's on rock pool communities with some invertebrates, they showed that if you look at more and more complex communities, starting with very unrealistic, simple experiments, going to more realistic field, real communities, you basically see start with positive relationship and end up with nothing if it gets closer to reality. Well, most mentioned issue after unrealistic assemblages is effect of species pool, because what's usually shown in BF experiments is some diversity. You may remember from the grass 1, 3, I don't know how many, ending with 60 species, but this is not reality. This is usually sown species diversity, which is there sown at the beginning of the studies, and it's then used throughout the whole time. However, if you look at not the wished diversity or sown at the beginning diversity, but realized diversity, you can see something what here Šuspa with Terka Richtecká, formerly Stachová, did here theoretically. So if you have species pool of few species, let's say 10 species, and look at the diversity productivity relationship, you see something you would expect, positive relationship, fine. However, if you look at, if you pick species from pool of 60 species, it's completely reversed. Why? Well, you can check that the axes are changing. Here it's shifted up, but basically not highly diverse communities. They are at the same levels. What's going up is low diversity plots. So diverse communities stays the same, but low diversity plots goes up simply because we are picking from bigger species pool, so the chance having their productive species is increasing, and it's causing exactly this. Some other authors showed exactly the same, and Hagan, already mentioned 2021, did exactly the very same thing. Well, this is initial sown diversity, this is what's shown typically, and he used realized diversity and looked at it within different species pools. And again, you see that if species pool is small, you get positive relationship. If it's big enough, you get negative one. So it's not effect of diversity, it's basically effect of a species pool you are selecting species from. Another issue can be that, and from my point of view, it's the most important one, that many dominants fails to persist. If you sow species from some species pool and pick them randomly, many of them simply fail because they are not used to be alone, or how to say it. Because what I see in a field, if you remove species and keep their dominance takes the place, and they are not the dominants just randomly. They are dominant because they have some specific properties. They are used to these specific environments, and therefore they are dominant, and so they can create, they can dominate the community. Well, if you sow there some rare species, it won't produce monoculture. This is nicely seen in Dietrich et al. 2020, which is again from MENA experiment. They did some nematode study, but used the plots, and they are showing that out of 60 species, that's the maximum diversity, the total species pool, 15 monocultures of these 60 species increased in productivity. Fine, this is what we would expect for all of them. If you remove all species from community except of this one, you would expect that it has a lot of space, no competition, so it will go up. But that's the case only for 15 of them. 45 species are either showing slight losses of their initial productivity or fail completely. 25% of these species fail completely in monocultures. Half of them is losing, so not having a good time, even they are alone, and just 15 are working like this. And this is hugely underestimating effect of low diversity plots, because if most of them die, then the low diversity plots are having low biomass. But it's not because they can't produce it, it's because you are sowing species that are not seen in nature alone, basically. However, there was a study of Jochum et al. 2020 showing that it's all fine, because they compared the results from BF experiments with, that's red dots, with black dots, which is some real field studies showing that the results are not differing if you compare them with real world studies. However, it's also questionable, because this is red dots are BF experiments, black dots are observational studies, so not experiments doing species removal in nature. This is observational studies. So. But anyway. So diversity has no effect on productivity, but what about stability? And again, in theory, if you have more species, world is better, so it's also more stable. More species, better stability. We look at this using just coefficient of variation. So basically, here you have on x-axis some ecosystem function. For us, again, it's biomass, and you look at, in time, how it's fluctuating, and that's what we are looking at the constancy here. So we are not looking at resistance or recovery from some perturbation, really at how the communities are fluctuating around some baseline. Surprisingly, there is not much conflict in literature in here. This is, again, a nice study of Hautier and plus 2 years old, showing that whatever the response is, which is different measures of stability here, and whatever the driver is, which is different measures of biodiversity, you see basically always positive relationship. So not much conflict in here. So we looked at this aspect again in the same community across seven years, and asked if, again, higher diversity mean higher temporal stability of biomass. We were also trying to disentangle mechanisms which are behind this stabilization, but I'm not going to show it, because it would be for another presentation, I am afraid. So just the most simple result. Again, you have the same treatments here, monocultures, free species plots, and controls on the right side. And what you see that coefficient of variation of community biomass in time is higher for monocultures, and then it's basically the same. So it seems that finally biodiversity matters for stability in real world communities. However, already 3 species are enough to stabilize the community to the very same level as 40 species in controls are able to. So monocultures are unstable, but the rest 3 species plots are already stable enough. So, how to conclude? It's a big mess, and I don't know. We see different results from observational studies, different results from biodiversity experiments. Maybe there is a way how to connect this via a species pool, as Hagan was recently suggesting, but this is maybe the way forward. So I don't know why it's so different, but I was at least trying to somehow give you some hints why it can be, but of course, apparently, you see that I am not completely independent. What we see in our experiment that diversity matters less than expected, but seems to be good at least for long-term stability in communities, and everything is in our experiment driven by dominance. Dominance can maintain basically whatever you look at quite well. You don't need many species. So what does this mean from conservation point of view? I'm not completely sure if I wasn't too, how to say, too controversial, or I was not trying to say that diversity doesn't matter. It, of course, matters for many aspects, but not specifically for above ground or below ground biomass, but this is a lot of studies claiming that it matters. So from my point of view, it doesn't matter for this particular function. However, there is many more functions, and there are already some people showing that rare species can maintain, can support some other functions than above ground productivity. So this is the way also forward to look at different functions. And from my point of view, issue is also that some studies are trying to somehow claim, not always directly, but indirectly, that if you have higher diversity, everything is better, but the same applies if you add invasive species into your community. At least in the short term, you are increasing diversity, productivity will be also higher. Remember the study of Naem, putting there invasive species, getting higher productivity, and this is maybe not better word. Of course, if I would sow Herakleum into ohrození, I believe it would grow there, it would increase diversity at least for a few years, and it would produce more biomass, but I don't know if it's exactly what we are looking, what we want to do. Well, actually, I want to do this, but that's another story in future. Let's see. So, what to do? Maybe we should also try to scale up biodiversity ecosystem research, not to be on small localities, and there are already a few studies trying to do this, at least theoretically. So it's comparable with observational studies. We need to look at more functions. We need to also look at more trophic levels, because most of the studies are looking at plants. Some studies are looking at invertebrates, bacteria, fungi, but I haven't found many studies that would try to connect all these together to look at the original meanings of stability, which was defined on the food webs, actually. And then finally we could maybe somehow connect these two opposing views on the very same relationship. So with all the disappointment, I thank you for your attention. I thank specifically Shuspa for leading me for all my years in his group, and many thanks to many other people who has ever helped me with what I've been doing. So thanks.
Thank you, Aleš, for a very nice talk, and now is the time for questions. Yes, Gonzalo.
Thanks for this broad presentation of the topic and your own work. I would like, well, a very specific question first, and if I can later a more broader one. First, when you were presenting the below ground study, you talked about, I think it was you talked about traits, but I haven't seen functional.
Oh, sure. Here. Sorry, I was skipping a lot of things we did actually there. I was trying to focus really on the line diversity, productivity, and ignoring everything else someone else did and we did. So we checked the traits, and again, you have here effective species richness on x-axis, different layers, so 5 layers. 0 to 5 centimeters, 5 to 10, 10, 20, 30, 20, 30, 30, 40. And the different traits, and you see basically no relationship at all.
Okay, I thought that you would contrast functional diversity to productivity. This is, again, taxonomic diversity against some traits.
We didn't look at functional diversity. You mean effect of functional diversity on below ground productivity? I was thinking about this, but it's we would need to distinguish individual species below ground, and we have just the total community characteristics. So we don't have information on a species level because we would need to do some molecular expensive stuff with. So would be nice, but we don't have species identity of individual roots. This is just at the community level always. So the summary across all the species in the community. So hard to calculate. But it would be probably very similar, because by creating diversity gradient in the experiment, we also created gradient of functional diversity, which you can see here, and it's very well correlated with the diversity gradient. So that would be, I don't know, I'm just speculating, but I expect that it would be very, very similar. And you had another question? Oh.
Yes, so you showed the effect of different plant size in biodiversity ecosystem functioning experiments. Are there any studies that selected species for the experiments based on traits like height or leaf area? Or would it be possible, if such an experiment is established, to somehow take species from the species pool, look at their traits like height and leaf area, and model the possible outcomes just based on the size parameters of plants, and compare it with the results from the experiment?
That's one option, and there is one paper, I don't have it in a presentation, also from an experiment where they put all species based on many traits, I don't remember which ones, but a lot of traits into some multivariate space, and then pick different corners and did communities composed of these corners of functional space. So they have treatments with as distinct functional composition as possible, keeping the same number of species or something like that. And I don't remember results, but something similar was already done. But sorry, I don't remember what was the result exactly.
Because in such experimental setting, I would expect maybe less bias due to different traits of plants.
Yeah, I think that most of the things we are actually looking at is driven by the dominant species, which has specific characteristics. And interesting thing would be to go to different communities, keep leaving their different dominant, because all communities are dominated by different dominants, and checking if it's always the dominant doing the job, even though if it's a different context, or if it's really based on our specific situation where Molina cerula is somehow weird compared to other species somewhere else. But that's something to be tested. But I would bet that dominance from the definition would be able to dominate communities even somewhere else then. But we are limited to single locality. That's an issue. Yeah. I'm not sure if I fully understand. You mean that in more productive environments you would somehow standardize the biomass between communities by what?
By the average size of the different species, so you can compare between systems that have different species composition.
Yeah, but there would be a problem that you don't have overlap with species identity. So I don't know if you would do it on a basis of height, then some plants can be quite a lot productive, although they are very low, and it also depends on phenological phase. If you, for example, imagine, I don't know, Betonica, on spring it's relatively low, but producing a lot of biomass compared to other species, then it grows, it's quite high. I don't know, but maybe I didn't get the question. That's now what came to my mind, but I'm not fully sure if we need this standardization.
I'm just trying to figure out if we have one very big, very dominant species that highly increases the productivity in the system, it is worth comparing to a system that has 15 much smaller species, because one will have much larger biomass than the other one despite the biodiversity, but this is inherently linked to the growth of the plants.
I believe this should be somehow mitigated when you look at the broad scales and have a lot of communities, or if you do it within single community, it's not a problem. But if you would compare 2-3 communities, then it would be tricky from the point you are raising, and probably also that the species composition is so different that hard to judge what's the driver. But then you have to somehow go into detail and look at what's doing dominant, what's doing rare species, and so on. But, yeah, I don't know. Thanks.
Okay, maybe I will have a question because I really liked your result with the comparison of České Budějovice region and Železné hory, where you actually showed that on local scale there was a negative, at least in Železné hory, there was a negative relationship between diversity and productivity, while when the two scales were combined, it was positive. Yeah?
Mm-hmm.
It's quite interesting, maybe, and also it was obvious that in Železné hory there is a larger species pool, yeah, and basically larger productivity and so on. And I was wondering whether this kind of study shouldn't be done somewhere where the diversity is saturated, yeah? Where actually you have like some communities which are like most species rich, and you also have a gradient of species richness, and then actually you could make a conclusion. Yeah? So you should go to White Carpathians.
Yeah.
I can imagine that there, if you did a study like that in White Carpathians, you could get a universal relationship.
Yeah, that's possible, and that's what Hagan is suggesting, focusing on species pool. One way is to go to localities with the same species pool, as you say, saturated would be nice, and then you can compare, or compare instead of species diversity, because the realized diversity in Budějovice region is lower because these are relatively intensively managed grasslands. In Železné hory there is more species because it's more, let's say, traditionally managed. So, but the species pool, we don't know what's the species pool. Depends how. But imagine if it's the same species pool. If it is, you still get different realized diversities, and that's what's compared, and it's not a good way. So maybe it's a good way either to go to the same level of species pool, but then you have questions how to define species pool. That's another story, or compare species pool, not species diversity.
Yeah, but I mean, in the trick between in the comparison between Železné hory and Budějovice is that in Železné hory you have a higher heterogeneity of the landscape, and also Železné hory is in the contact with thermophytic pools. So there are quite some thermophile species occurring at some sites which you don't have in the Budějovice basin.
Sure.
So it's not only about the management.
We were not trying to show what's the relationship. We were trying to look into two different regions with two different management histories and show it's context dependent, and we shouldn't do what we did.
Yeah.
But we did it for showing that no one should do it again because it's apparently very different, even within the very same habitat type, temperate grasslands, which is in these global studies completely negligible detail, but even within the very same habitat, you see completely opposing results. Just because, and that's our interpretation, we don't know, but we think it's just the different management strategy, mostly, or species pool, yeah, who knows. Sure.
Than just the statistical correlation between species and productivity, which in the end it's comparing like apples with oranges, because there are tons of possible effects there and thousands of confounders.
Yes, you have a lot of evidence from artificial BF studies showing consistently positive relationship, but You don't have so many field studies because it's harder. We have small experiment because you have to go there several times a year and weed in individuals from the plots to keep it, which is another aspect. Maybe if we wouldn't keep the diversity low intentionally, maybe it would look differently. That's another story. So the thing that there is a lot of evidence accumulated over years that it's positive may simply be wrong, as already several times criticized, because it's based on a premise that you can select species randomly. And another thing, which is actually relatively funny, you mentioned complementarity. It's usually connected with complementarity and selection effects, and there is a very interesting recent paper here trying to look at this partitioning method you probably know about, which started with Floro and Hector 2001, and that there are another advancements in this partitioning, and they tried to somehow critically comment on these different approaches, how to disentangle the role of complementarity and selection effects and sort it into different types of partitioning, let's say. So first type is this Loro and Hector equation. Most of the studies are using they put it on an axis, degree of incoherence. This is trivial, this is nonsensical. So their equation is here on a trivial part, which is mathematically correct, but saying nothing. And that's what most of the studies are using. This equation, nice, mathematically saying nothing. And then there you have some foxes. It's very commonly used, some foxes, the derivations from this. So this is type 2 biodiversity, bookkeeping, or spreadsheet formulas which involve trivially correct use of arithmetic. Fine. And then you have these mathematically nonsensical formulas which are also used, and then there is another type, improper partitioning schemes. So basically, all the schemes that by you can recently partition the biodiversity effect into these components is based on these authors, not based on me, based on these authors either trivial, so saying nothing, or nonsensical. And I like this citation from this work. The price equation might be generally thought as of being useful just because a significant fraction of ecologists doesn't care. So thank you for this question. I like this paper, and I.
That paper is a debate between authors, and they are.
Yeah, you know it. So it's generally funny paper. I recommend reading because you will be laughing half of the text. It's funny. They are very.
Yeah, in your experiment with weeding, you weeded non-dominant plants above ground, but what about below ground? So the plants remain there.
Well, we are trying to pull it out with roots, but of course some species are easier to be removed, some are harder, potentially. Yes. So we believe we are able to kill them relatively sufficiently after many years. Second year it's questionable. After more years we believe, but we don't know because we don't see what's below ground. We believe that it's all right. And this is the weeded biomass. You see what was weeded at the beginning. It's a red bar in different treatments. So, of course, in monocultures, we weeded quite a lot in the first year, but the second year it was just small portion of it, and it stays basically stable across across all treatments. So the first weeding was really intensive. You have seen also the picture where the plot was half empty, but already next year. And this is cumulative biomass from 2 weeding events. So if you, it's actually doubled if you would look at just single weeding event. And this is from 1 meter plot, and what we are usually working with is square, which is within this 1 by 1 meter plot to avoid edge effect, which is another reason why we believe that even below ground the management, the treatment is working, because there is a buffer zone. So we are hoping that the plants from around are not reaching to where we really look at the.
So how long does it take to weed one such plant?
At the beginning, we did it for 10 days with 3 to 8 people, let's say.
How many plots?
20.
Okay.
Well, 25, but the disturbance control is quicker because you don't need to. It's quite demanding on your brain, because monocultures are fine. You are keeping there single monoculture and weeding the rest. But if you have 12 species plots, you need to keep in mind that you are keeping 12 species, and in each plot it's different 12 species. So it's a little bit crazy, but.
Okay, and technically, when did you do the weeding?
Usually May.
So in May. So
In the middle of May.
and then you sampled the biomass
After a month.
after months, yeah, and then again like
August.
yeah. Okay.
End of August.
It might be interesting to make a metagenomic analysis of the below ground part.
Sure.
It's not that difficult nowadays.
We have a lot of ideas, and we did a lot more things that I was actually showing. Now that we are about to submit one more complex paper where we look at the different fluxes of carbon. So we are measuring net photosynthesis, respiration rate, below ground biomass, above ground biomass, decomposition rate. What else? We have litter there and few other things, and we are looking at all this stuff somehow more complexly, but it would be another hour presentation.
It would be nice to see kind of like multifunctionality. For instance, I can imagine that nectar production or pollinators would actually show quite strong dependence.
I think it would show nothing, because it's a really small scale. It's 1 meter plots next to each other. So I'm not sure if we could see something with.
Yes, because Molinia doesn't produce any nectar, while
Okay.
some forbs which you have there for sure actually.
But it would be interesting if it's not producing anything. Well,
it is.
I would do it across multiple localities. That would be more interesting, I think, because.
That with some other parameters than productivity, you could actually some other biodiversity, ecosystem functioning parameters, you could get some relationships.
Yeah, that would be nice.
So, any further questions? If there are none, then actually we can still continue the discussion in the path. There is